Hello everyone, welcome back. This is the fourth section of the lecture Heart and Embryological Origin. In this section, we'll be talking about the molecular regulation of what actually contributes to the formation of adult heart. This section has been further divided into three subsections. We'll be talking about the heart forming region and what genes and what factors actually act on this region to form the adult heart tube. Then we'll go into the details of retinoic acid and how it helps in the formation of venous components of the heart and the cardiac septation, what genes actually play a role in cardiac septations. Then we'll see what factors affect the lengthening of the outflow tracts. So now the first subsection is the heart forming region. For the heart forming region, you need to keep in mind three of these important factors. The first is the transcription factor NKX 2.5, this one. Then you need to know BMP, the bone morphogenic proteins 2 and 4. These are the bone morphogenic proteins. And lastly, we have WNT inhibitors. These are the WNT inhibitors. So what actually happens, the transcription factor 2.5 is being expressed, is being induced in the heart forming region for the progenitor heart cells to begin the formation of heart. For the induction of NKX 2.5, we need BMP 2 and 4. BMP is the bone morphogenic protein that needs to be present in the embryo so that NKX 2.5 can act. At the same time, the activity of WNT proteins need to be inhibited. So let's see how all of this happens. So the anterior endoderm induces the splanchnic mesoderm to form a heart-forming region. We talked about this before. The splanchnic mesoderm is very important when it comes to formation of heart. So the anterior endoderm is now inducing the splanchnic mesoderm. And how is it inducing the splanchnic mesoderm? By inducing the transcription of NKX 2.5. NKX 2.5 is not an acronym but simply a transcription factor. Now, it requires the signals for the secretion of BMP2 and 4. BMP is bone morphogenic protein. So the transcription of NKX 2.5 requires the presence of bone morphogenic proteins 2 and 4. At the same time, the activity of WNT proteins must be blocked. So the WNT proteins, which are usually acting within the embryo, have to be blocked for the activity of NKX 2.5. So where do these blockers come from? These inhibitors are produced by the endodermal cells which are adjacent to the heart forming region. So these inhibitors are produced which finally inhibit the WNT proteins. When WNT proteins are inhibited under the influence of BMP2 and 4, there's induction of NKX 2.5. So retinoic acid. This figure represents the structure of the retinoic acid. I'm sure you must remember some of it from your chemistry. So what does the retinoic acid do? Retinoic acid helps in the determination of the venous portions of the adult heart. Now let's see how that happens. So the mesoderm, which is adjacent to the primitive sinus venosus and the atrial regions, it secretes the retinoic acid. So towards the caudal end of the heart tube, the mesoderm, which is adjacent to the caudal end of the heart tube, it secretes retinoic acid. Exposure to retinoic acid specifies the venous portions of the heart tube. Since the mesoderm surrounding the caudal portion tends to secrete the retinoic acid, so the caudal portions are bound to form the venous portions of the adult heart. So they express the retinyl dehydrogenase genes, which makes them able to form their own retinoic acid. It commits them to becoming the caudal heart structures and hence the venous heart structures. So now let's talk about cardiac septation and what factors help in the formation of cardiac septa. Septa are actually the divisions within the heart. We talked about this. Initially, the heart tube consists of a common atrium and a common ventricle. However, in the adult heart, we have two atria, two ventricles, or they are separated from each other by cardiac septa. We also have the outflow canals, the aorta and the pulmonary arteries, which are also separated by the cardiac septation. So the factor that that the gene that plays an important role in cardiac septation is TBX5. TBX5 is a transcription factor which contains a DNA binding motif which is known as the T box. So it is expressed later than NKX 2.5. So initially, the heart-forming region has to express NKX 2.5 with the help of bone morphogenic proteins 2 and 4, and by inhibiting WNT proteins. These inhibitors are secreted by the endoderm, which lies near to the adult heart-forming region. After that has been formed, the heart tube has been formed. Now, the heart tube has to be there for the process of cardiac septation to take place. So, in the process of cardiac septation, the gene TBX5 plays the most important role. It is actually a transcription factor with the DNA binding motif, which is known as T box. Here in the figure, we can see various defects. So, this is the clinical picture of what happens if the TBX5 is not expressed clearly. In type 1, we can see here a large common ventricle has been formed. Here in red, you can see a large common ventricle, and the ventricle septation is not there. In type 2, a large left ventricle has been formed and a small right ventricle has been formed. Both the outflow tracts are being originated from this large left ventricle. In the third type, we have a large ventricle, a left ventricle, a smaller right ventricle, and this aberrant structure. So the structure of the heart does not remain the same if there is a defect in the TBX expression and the septation does not take place as it is supposed to be. So we have already talked about the process of cardiac looping in detail. Now let's take a look at various mediators that act at the process of cardiac looping. So it depends on the laterality pathway. Laterality pathway is very important as it helps to develop the axis of the embryo around which all the organs are being developed. It also depends on the expression of PIT-X2 in the lateral plate mesoderm on the left side. So again, lateral plate mesoderm is very important when the development of heart is being concerned, and the expression of PIT-X2 in the left side helps in the determination of helps in the formation of correct cardiac loop. So how does it do that? It helps in the deposition and function of the extracellular matrix molecules in the process of cardiac looping. Now we have a role of NKX 2.5 as well, which upregulates hand 1 and hand 2. The upregulation of hand 1 and hand 2 helps in the looping of the heart and expansion and differentiation of ventricles. We saw that individually the heart tube is a thin single line structure. However, with this upregulation of hand 1 and 2, it helps in the looping of the heart as well as expansion of the heart tube to form the adult heart structures. Now moving on, let's talk about lengthening of the outflow tracts. So the lengthening of the outflow tracts is by the secondary heart field, and it is regulated by the sonic hedgehog gene. Sonic hedgehog gene is a very important gene which is being expressed throughout the embryo and plays a lot of roles. One of the role is the elongation of the outflow tracts. So these are the outflow tracts. These are the aortic arches being formed. This is the third, this is the fourth, and this is the sixth aortic arch, and this tends to be the dorsal aorta. This is the developing portion of the conus cordis that helps in the formation of the aorta, and this is the developing pulmonary trunk. So we are talking about the lengthening of this section of the heart now. So these are expressed, the sonic hedgehog gene which acts on this area is expressed by the pharyngeal arch endoderm. So a very important structure. It acts through its patched receptor which is expressed by the cells of the secondary heart field. So the sonic hedgehog gene, which is supposed to act on the outflow tracts to help in their elongation, is expressed by the endoderm of the pharyngeal arch. So the pharyngeal arch endoderm, the pharyngeal arches here, their endoderm expresses this sonic hedgehog gene. And how does it act? It acts through its patched receptor which is present on the cells of the secondary heart field. Now what does they do? This stimulates the proliferation of cells and hence the elongation of the outflow tracts. So notch signaling is also responsible for regulation and migration of the differentiating neural crest cells. So these are the neural crest cells. They are migrating and differentiating into the aortic arches. And what is responsible for that? These are the notch signals. They cause septation. So these neural crest cells help in the septation of the developing aorta and septation of the outflow tract, both the aorta and the pulmonary trunk, and they help in the formation of the aortic arches here. You can see these blue, these are the neural crest cells. They are penetrating the aortic arches, and then the developing outflow tracts, which include the developing aorta and the pulmonary trunk. They help in the formation of septation in these structures as well. So this was all about the molecular regulation. In this section, we saw how the heart tube loops around each other to form the adult cardiac structures. We saw how various agents, various transcription factors acted on the process of looping, and what was important in the process of looping, and how the interaction between these agents contributed to correct formation of the adult heart. I hope you enjoyed this section. For further sections, keep watching sqadia.com.